Hello, learners. Welcome to Biotechnology Lectures, and I'm Dr. D. Prabhavati, here to brief you on the structural features of DNA. DNA is deoxyribonucleic acid, and as we know, it's a genetic material that codes for the information of all the life processes, inclusive of the growth, replication, metabolism, etc. And it is present in all the cells except a very few exceptions like the virals, RBC, the C cells. Otherwise, DNA is a genetic material which is present in all the cells and it is passed down from one parent to the other. And structurally, it is a polymer of deoxyribonucleotides. Let's have a brief introduction to the historical events which led to the discovery of DNA structure. In 1869, Frederick Mencher identified a substance in the nuclei of the human white blood cells which he called it as a nuclei. In 1944, Erwin Chargaff suggested few rules for the arrangement and the composition of the DNA. 1952, Rosalind Franklin's X-ray diffraction studies of DNA was a milestone which led to the discovery of the DNA structure. And the credit goes to Watson and Crick, who discovered the double helical model of the DNA in the year 1953. To begin with, let's look at what its Chargaff rule is. This is the rule which led to the fundamental principle that provided the insight to the structure and behavior of the DNA, and it also outlines a specific base pairing rule between the four nucleotide bases in DNA. And this formed the basis of the DNA's double helical structure. Now, what does this state? In a DNA molecule, the amount of adenine equals to the amount of thymine, and the amount of cytosine equals the amount of guanine. In any DNA molecule, the ratio of adenine to thymine and cytosine to guanine is approximately equal. Let's come back to the base composition a little while later. Now, let's look into the next historical event which led to the discovery of the DNA. Rosalind Franklin did X-ray diffraction studies on the DNA. She passed on the X-ray beam and studied the shadowed structure of the DNA. And from this photograph, the famous photo 51, Watson and Crick inferred three major structural features of the DNA. One, the DNA is helical. Two, the helix is double-stranded. And three, the distance between the base pairs. So they inferred these three features from the data of Rosalind Franklin and the base composition of the Chargaff's rule. Having said that DNA is made up of nucleotides, let's have a look into the components of the nucleotides. So the nucleotides have major three characteristic components: a nitrogenous base, a pentose sugar, and a phosphate. The molecule without the phosphate group is called as a nucleoside. That is, a base and a sugar is a nucleoside. A base, sugar, and a phosphate is a nucleotide. So these nucleotides are joined together to form two long strands and that twist to form a structure called a double helix. Now, these are the bases which make up the DNA. DNA is made up of purines and pyrimidines. The purines are guanine and adenine. The pyrimidines are cytosine and thymine. So guanine pairs with cytosine, adenine pairs with thymine. The two bases, adenine pairs with the thymine with two bonds, hydrogen bonds, and guanine pairs with a cytosine with three hydrogen bonds. So it's a rule that a purine bonds with a pyrimidine and a pyrimidine bonds with a purine. So a base pair basically contains a purine and a pyrimidine. Now this is a structural feature which depicts the base pairing. Thymine always pairs with adenine with two hydrogen bonds, and cytosine pairs with guanine and this is held together by three hydrogen bonds. Apart from the nitrogenous bases, DNA is made up of a deoxyribose sugar. The deoxyribose sugar lacks a hydroxyl group on the 2' carbon. So hence its name deoxyribose sugar. And the 1' carbon of the pentose sugar bonds with the nitrogenous base and the 5' end carbon bonds to the phosphate group. Then the phosphate group bonds to the 5' end of the nucleotide. A phosphate group A phosphate group attaches to the nucleoside. Earlier, we discussed the pentose and the base makes up the nucleoside. When a phosphate group attaches to the nucleoside, it forms a nucleotide. So this phosphate group is a backbone for every single strand of the DNA molecule. To detail the structural feature, now we can find that this is a structure of a nucleotide, adenosine monophosphate. So the major component here being the base held together by the deoxyribose sugar. So the sugar, the C1 position and the nitrogen bases are held together. And the 2' end, as you can see, lacks a hydroxyl group. The 5' end, we have the phosphate group attached to it. So this makes up the phosphoester linkage, and this one is a glycosidic linkage. So the DNA involves multiple bonds like the hydrogen bond between the bases, the glycosidic linkage between the base and the sugar, and the phosphoester linkage between the sugar and the phosphate. So let's start with the Watson and Crick model of DNA. So as we have been discussing, the DNA molecule comprises two individual strands of polynucleotide molecule which are linked together by a hydrogen bond. Each base of the nucleotide pairs up with another. Purine bases always pairs up with the pyrimidine bases. The double helix is right-handed. It is in a clockwise direction and completes one turn at a distance of 34 angstroms. The DNA strands are anti-parallel to each other. That is, if the starting point of one strand is 5', then the starting point of the another strand is 3', and vice versa, where the nucleotides link together with the sugar group of the 3' and 5' end, which is due to the polarity of the phosphodiester linkage. The additional stability to the double helix is provided by the base pair stacking. Now let's have a detailed illustration of the DNA structure. So as we can find, the bases are held together by the hydrogen bonds. This is on a one strand. This is a single strand and this is another strand. Now these two strands are held together by the hydrogen bonds between the bases. As we can see here, the CG or the GC base pairs are containing three hydrogen bonds and the TA is bonded with two hydrogen bonds. The bases being bonded by the hydrogen bonds with the sugar the sugar of the consecutive bases are attached to each other by phosphodiester linkage. So here we can find that the 5' end has a phosphate group which is free. The 3' end again contains a phosphate group which bonds to the 5' end, the phosphate of the next incoming nucleotide. So this strand runs in a fashion like 5' to 3'. Since this 5' end is free, this is a 5'. It runs from 5, and here as you can see the 3' is free. Whereas in between the 5' and the 3' are involved in the ester linkages. And similarly, to the opposite, we find that the next strand runs from 5' to the 3' in anti-parallel direction. So as we can see, the sugar phosphate makes up the backbone, whereas the bases are found within the sugar phosphate backbone. And to sum up again, there are three major bonds which are involved: the hydrogen bond between the bases, the glycosidic bond between the base and the sugar, and the phosphodiester linkage. The helix contains 10 nucleotides in each turn with an internucleotide distance of 3.4 angstrom, which we call technically as the base pair rise, the distance between two base pairs. Whereas the full turn helix length is 34 angstrom, that is the distance between two turns, which contains 10 nucleotides, isn't it? And the diameter of the total helix is 20 angstroms units. This helical turning of the double helical DNA forms two grooves, the major groove and the minor groove. The major groove appears deep and wide and is a specific binding site of the proteins, whereas the minor groove is the distance between the two strands. So to sum up the four major features of the Watson and Crick model of the DNA, the sugar phosphate backbone is on the outside. The sugar phosphate backbone is on the outside and the bases are inside. The strand is held together by hydrogen bonds. A only pairs with T and G only pairs with C, which we call it as a complementary base pairing. And the two strands run in anti-parallel direction. And the DNA twist into a double helix, which are held by more hydrogen bonds, leading to the formation of the major groove and the minor groove. So the pitch of the DNA, that is the length, the total distance for one complete turn is 34 Armstrong units, which contains approximately 10 base pairs, and the distance between each base pair is 3.4 Armstrong units. And the total diameter of the helix is 20 Armstrongs. So, you understood the basic structural features of the DNA. Join with me for more lectures on the basics of molecular biology. Thank you.